All right, so now I want to change directions for a minute and think about what happens when DNA replication goes wrong. So there are lots of reasons why there can be errors in DNA replication. So it's rare, but polymerases actually can make mistakes. They can insert the wrong nucleotide during DNA synthesis. So that's one way that errors can happen. But also, there are lots of mutagens in our environment that we're exposed to that are damaging, right? So we know that cigarette smoke is associated with lung cancer because of the mutagens that are present in smoke. Many pollutants in our environment can be harmful to human health because they can cause mutations. Too much sunlight can lead to skin cancer because it causes mutations in the DNA. And in addition, in the South, our favorite grilled meats that we all like to cook out during the summer, that charred meat on the edges, that black stuff, is actually the same carcinogens that you can find in cigarette smoke. So that's also another cause of cancer. So there are lots of carcinogens in our environment. We're exposed to them all the time. However, luckily, our body has mechanisms to repair the damage that's caused by these types of mutagens. We're going to discuss 3 of these mechanisms. The first one generally repairs errors that are made by DNA polymerase, and that's DNA polymerase proofreading. So essentially what happens during proofreading is that DNA polymerase is synthesizing the new strand of DNA, and it inserts an incorrect nucleotide. So you have a mismatch here. Now this will actually distort the helix, causing sort of a bump in the DNA helix. When that happens, most of the time DNA polymerase will recognize that error and will proofread by backing up to take out that incorrect nucleotide using the 3' to 5' exonuclease activity. So remember, we're moving in the 5' to 3' direction synthesizing. So the exonuclease activity goes back in the opposite direction to remove the incorrect nucleotide, and then DNA polymerase starts forward again. So we know that this proofreading activity is really, really important because normally after replication, there's about 1 mistake per 1 million base pairs when polymerases have proofreading. However, if we remove that proofreading activity by replacing DNA polymerase with a mutant enzyme that lacks the 3' to 5' exonuclease activity, there's actually a mistake for every 1 in about 1,000 to 10,000 base pairs. So a really huge increase in the mutation frequency. So that's one mechanism of DNA repair, of repairing these types of mutations. Another mechanism is called mismatch repair. Generally, mismatch repair happens when the DNA has been modified in response to some kind of mutagen. So you have a mismatch because one of these bases has usually been damaged. It also can happen if a mistake was left in after DNA and the DNA polymerase proofreading activity missed it as well. So during mismatch repair, remember this mismatch causes a distortion in the DNA helix, and these repair enzymes recognize this mispaired base, and essentially they cut out a very short section of DNA, and then a DNA polymerase, a very special DNA polymerase, fills in that gap, and DNA ligase will seal any remaining nicks. So essentially mismatch repair just removes a very short segment of DNA and repairs the mismatch. So we also know that this pathway is very important because there are humans born with mutations in these mismatch repair proteins, and this leads to a syndrome known as hereditary non-polyposis colon cancer. People who are born with mutations in this pathway generally develop lots of tumors in their colon, as shown here, as well as they're susceptible to several other types of cancers, such as ovarian cancer in women. So mismatch repair is clearly very important for removing damages, particularly in certain target organs like the colon. So our last type of repair is nucleotide excision repair. And nucleotide excision repair most commonly removes really large distortions in the DNA. One of the most common distortions that it removes is called a thymine dimer. So when you have two T's that are next to one another in the same strand, when those are subjected to UV light, for example, when you go out in the sun, that can cause these two T's to bind to one another as opposed to their match, to the A that they should be matched with. So this thymine dimer, because these thymines are now cross-linked to one another, creates a really large distortion in the DNA helix. So nucleotide excision repair proteins recognize this really large distortion in the helix, and similar to mismatch repair, they remove a segment of DNA. This segment is actually thought to be a little bit longer, more like between 12 and 14 nucleotides. It's actually endonucleases that clip here within the strand of DNA to remove this damaged DNA. Then DNA polymerase is going to fill in the gap, and DNA ligase is once again going to seal the nick. We also know that this pathway is very important because of known human mutations. So there's a disease called xeroderma pigmentosum. So children who are born with this disease are missing this nucleotide excision repair pathway. And children with this disease have a 10,000 times increased risk of skin cancer. So here you can see the damage that this child has sustained from just going out in the sun. And in fact, they've developed special sun suits for kids with XP. And for any of you who have read the Odd Thomas books, those books by Dean Koontz, those books are actually about a guy, Odd Thomas, who has xeroderma pigmentosum. So that's your factoid for the day. So those are our three major DNA repair mechanisms: DNA polymerase proofreading, which fixes errors made during DNA replication; mismatch repair, which fixes small mismatches in the DNA left after replication has occurred; and then nucleotide excision repair, which repairs larger DNA damages. So now I want to move on to talking about the end problem in DNA. So first, let's consider what is the end problem. And of course, this is unique to eukaryotes because prokaryotic plasmid DNA is circular and there is no end. So this is unique to eukaryotic chromosomes. And what scientists noticed is that these eukaryotic chromosomes actually shorten over time. So for example, Dolly the sheep, because she was cloned from an adult animal, she actually had much shorter chromosomes than a normal sheep who had just been born. So let's take a look and consider why this happens. So let's imagine that we've zoomed in on this replicating strand. So here we have our old strand is shown in dark blue, and our new strands are shown in light blue, and the RNA primers are shown in red. So on the lagging strand, after we remove this RNA primer, it leaves a gap. And remember that DNA polymerase cannot do de novo synthesis. Only primase can. So that means that DNA polymerase can't just start here and fill in this gap. So you have an end where you have just single-stranded DNA where the DNA essentially hasn't been replicated. So now let's consider that we go through a second round of replication. These two strands are going to separate, and now they're your old strands, so shown here in dark blue and in light blue. So now this strand actually made a shorter chromosome. And as this goes on, essentially you get shorter and shorter DNA molecules over time. So there has to be a mechanism to deal with this in eukaryotes, because otherwise the chromosomes would eventually get shorter and shorter, and you would start losing really important pieces of DNA. So the mechanism that takes care of this is called telomerase. So telomerase is an enzyme that adds repetitive DNA caps at the ends of eukaryotic chromosomes. So here you can see it actually, within this enzyme, it has a piece of RNA that it uses as a template, and it adds this TTAGGG, repeat over and over and over at the end of the chromosome. And this gives a piece of DNA that doesn't really code for anything, so it doesn't matter if it gets shorter with time, so that those chromosomes can shorten over time. But something important to know about telomerase. In humans, telomerase is only active when you're an embryo. So your telomeres are as long as they are ever going to be when you're born. That's it. That's how long your telomeres are. So when your telomeres run out, then you start losing important pieces of DNA that code for protein. So because of that, human cells undergo senescence, meaning that they stop dividing. The maximum number of cell divisions is thought to be about 70 cell divisions. So it's thought that telomeres may actually be the limitation that limits the human lifespan. So you're probably all thinking, well, why don't we just express telomerase? Then we would all live forever, because that would be awesome. Except here's the reality. There are cells in your body that overexpress telomerase, and they're immortal. They're called cancer cells. So unfortunately, turning on telomerase when you're not supposed to can promote malignancies. So guess we won't be living forever just by expressing telomerase. Okay, so the last thing I want to talk about is DNA packaging. So we've already talked about the DNA double helix, but I want to talk about how this DNA is packaged tighter and tighter to get to this condensed chromosome stage. And of course, if you're not in mitosis, you're really somewhere along this pathway, but I want to talk about how this DNA is compacted. So from this part, make sure that you know the proteins that drive the different levels of DNA compaction. So our first level of DNA compaction is the nucleosome. So the nucleosome is simply 2 loops of DNA, it's about 200 base pairs, spooled around a histone octamer. So here, the histones are globular proteins that can bind DNA. So the octamer part means that there's 8 of them. So here you can only see 4, but there would be 4 on the opposite side as well. So you have 8 proteins, and 2 loops of DNA are wrapped around each one of these histone octamers. And together, that forms the nucleosome. So each one of these units is a nucleosome. So now those nucleosomes can be compacted further into the 30 nanometer chromatin fiber. And how that occurs is that one of these histones that we showed here below, one of these histones actually has a tail that sticks out. This histone H1 tail can be used to cluster nucleosomes into this 30 nanometer chromatin fiber. So our next level of compaction is the 300 nanometer fiber. And essentially here what we have are scaffolding proteins that create these loops. So the final level is the condensed chromosome, shown here. So the condensed chromosome depends on these aptly named proteins called condensins. So condensins drive condensation of the 300 nanometer fiber into this condensed chromosome structure. So just to review, you go from the double helix to the nucleosomes, which depend on the histone octamer, to the 30 nanometer fiber, which depends on histone H1, to the 300 nanometer fiber, which depends on the scaffolding proteins, to the condensed chromosomes, which depends on the condensins. So that's the end of the video. So thank you for watching. I hope you took notes. Feel free to watch it as many times as you want. You will be held responsible for this material on the exam. Make sure that you've covered those learning outcomes that we noted in the second slide. Also, don't forget to do your replication bubble activity and bring it with you to class on Monday. I hope you have a great weekend.